is Dr. Amit Shradd, Department of Medicinal Chemistry, Naipur, Ahmedabad, Gujarat. And he'll be talking about targeting triple negative breast cancer with small molecular inhibitors of tumor pyruvate kinase M2. Over to you, sir.
Good afternoon, everyone. Myself, Amit Sharad, and I'm from the Department of Medicinal Chemistry, Naipur, Ahmedabad. And the title of my talk today is Targeting Triple Negative Breast Cancer with Small Molecule Inhibitors of Tumor Pyruvate Kinase M2. This is the target that my lab has been working upon for the last 1 decade. As the audience is quite aware that breast cancer, everyone spoke about it. It is initiated with the small lump being formed, and then the lumps grow, and if they are allowed to grow uncontrollably, they can metastatize and it can be very much fatal. And as far as talking about the incidence rate, Africa is having one among the highest incidence rate, followed by Asia and other countries also. And in India, the spread of the breast cancer is almost there in every state, in which Punjab, Delhi, Haryana, and some part of the south also are also at first place in case of the reporting of the incidences. Globally, if we talk, there are several million cases, but in India, every 4 minutes a woman is diagnosed with the breast cancer, and in every 13 minutes a woman dies of it. Out of every 4 women that are being diagnosed with breast cancer, one of them could have triple negative breast cancer. And as far as talking about the treatment modalities, there could be surgery, radiation, hormonal, or chemotherapy per se. And in the chemotherapy per se, several kinase inhibitors and other drugs also are playing quite important role. But straight coming to the topic, out of which pyruvate kinase M2, although there is no specific drug in the market targeting this kinase as of now, has hit the market against breast cancer, but it is emerging as one of the important target. And actually this is an glycolytic enzyme. If we talk about the location of this enzyme, it is a cytosolic in nature firstly, and it is present in 4 isoforms: PKM1, M2, PKL as well as PKR. While PKL and PKR are located in liver and RBCs respectively, M1 and M2 have been found, M2 predominantly, and M1 also has been found to be active in rapidly proliferating cancerous cells. So if you just look upon the location of this pyruvate kinase M2, if we just look into the normal cell as well as the cancerous cell, the fundamental difference lies here, that in case of a normal cell, phosphoenol pyruvate gets quickly converted into the pyruvate with the assistance of pyruvate kinase M2, which is by and large tetrameric in nature. But once this pyruvate kinase M2 is mutated and gets converted into the dimeric form, the phosphoenol pyruvate is not converted into pyruvate at all, rather it gets converted into the lactate, what we commonly call as the Warburg effect. And from here everything starts off. This is actually the point of intervention where we are working upon. We are working upon this much target, and we are trying to develop certain molecules which could convert this dimeric form to the tetrameric one, or could inhibit the dimeric form of the pyruvate kinase M2, which is located in the aggressively dividing tumor. While we were looking for several molecules that from where to start with, we stumbled upon several different molecular types and their structures. Dr. Patel is sitting there, he works extensively on heterocyclic chemistry. We stumbled upon one of the important nucleus that exists in the organic synthesis, particularly in the pharmaceutical sciences. If you look into this molecule, or this small 5-membered heterocyclic ring, this is thiazole. This thiazole ring has been found to be benign to the human biology also, because it is also one of the metabolically benign type of molecule, because it is also one of the constituents of vitamins. So when we looked into several anti-cancer drugs, we found that thiazole is having one of the predominant presence in several anti-cancer drug types. And after screening almost 21 different type of heterocycles and having extensive computational studies, I'm not showing the data because of the paucity of the time, We finally realized that we are going ahead with this molecule, and we designed certain molecules, and we quickly developed these molecules, converting pyrimidine molecules into chloroacetyl derivatives, and the chloroacetyl derivatives were reacted with several thioureas, leading to the formation of thiazole based imidazopyridines or pyrimidines. This was probably positioned over here because it could have some 9-membered molecule, like closely mimicking the purines and the pyrimidines existing in the body. So we quickly synthesized the library of these molecules, and after synthesizing and getting them in adequate amount, close to 45 different derivatives were synthesized. In good yields they were obtained. We quickly checked the pyruvate kinase activity of these molecules, how these molecules are behaving once we are testing them against the desired target or the target of interest. Some of them were found to be quite potent inhibitors of pyruvate kinase M2. So I am straightway coming to the findings that what we, I'm sorry, the slides are. We quickly evaluated the molecules, and the 3 parameters we set initially were in silico studies, in vitro studies, and in vivo studies. So I am quickly coming to them one by one. In silico studies, we found that the molecules that we had designed were showing ample interaction with the ADP binding site of the pyruvate kinase M2. It was having a very good binding score to the protein of the interest. And we quickly done other studies so as to know that what the fate of the molecule could be. We found that molecule was having its HOMO as LUMO closely located. In case of MESP studies, we found that molecule was having electrophilic sites. And it has been found in a generalized way that usually the anti-cancer drugs generate electrophile. A very simple and fundamental example I am giving you, for example, chlorambucil. Chlorambucil hydrolyzes to a carbocationic form in vivo, and the carbocationic form forms an adduct with the DNA, the N9 position of the DNA. In the similar way, the molecule was having an electrophilic center generated, which was probably interacting with the nucleophilic sites of the protein. The MD simulation studies showed that the molecule was stabilizing the protein. Then we quickly switched on to the in vitro studies, and the in vitro studies could help us to conclude that the molecule synthesized, which was 7D, which was a thiazole based molecule, was inhibiting the pyruvate kinase M2. We also did the studies in 2D cell culture as well as 3D cell culture. 3D cell culture because 3D mimics closely the physiological environment, and as we were planning for in vivo studies to be taken down the line. So we quickly did all these studies, and the results were quite convincing, and the molecule was doing pretty well. And compiling up the in silico as well as the in vitro data, we could figure out that some of the molecules were quite potent against a panel of cell lines and our protein of interest that we had interest into. And in the LDH coupled enzyme assay, the molecule was doing pretty well and showing a nanomolar range potency. And when they were evaluated in 2D and 3D, it was found that molecule was doing pretty well, and we were having closely a very good low range micromolar activity against 4T1 cells. This was followed by the cell cycle analysis, and we could figure out that our molecule was arresting the cell in the G2 phase of the cell cycle. The molecule was inducing apoptosis in 2D as well as 3D cells in a similar way. Further, to validate the data, the RT-PCR study determined that how PKM1 and M2 and VEGF, which is one of the angiogenic marker, are being inhibited after treatment, after giving the cells insult by our compound, which was 7D, and the data was quite found to be significant in almost all the cases. This was rapidly followed by in vivo investigation, where 41 cells were taken and they were injected into the memory pad of the nude mice, and when the tumors had grown up to 100 mm3, so then the animals were sacrificed. It was found that in the low dose, and in the low dose 7D, that is 25 mg per kg, the molecule was reducing the tumor to a very significant length, and in vivo, IHC examination in vivo hematoxylin also showed that after treatment there were very much less striations in the tissues, and in vivo immunohistochemical analysis also gave us positive results on Ki67 and VEGF angiogenic marker. To conclude, the rationally developed molecule, which was a thiazole based molecule, did pretty well and inhibited the pyruvate kinase M2. The pyruvate kinase M2 inhibitor, which was rationally designed, scored well in vitro as well as in vivo studies, but the initial onset point was in silico studies, and in silico studies gave us a clue that out of the 21 heterocyclic scaffolds that we were selecting and 1190 molecules that we were initially planning, we only synthesized 45 molecules, out of which one is potent enough. And the result of this study is just recently published in Journal of Medicinal Chemistry. Thank you very much for the patient listening. Thank you very much, chair and co-chair and all the learned audience. Thank you very much.